Armando Hasurungan, Biology and Medicine videos. Please make sure to subscribe, join the 4-man group for the latest videos. Please visit Facebook Armando Hasurungan. Please like, and here you can also ask questions, answer questions, and post some interesting things, including your artworks. You can also change the quality settings to the highest one for better graphics. In this video, we're going to talk about eucasinoids. Eucasinoids are made or generated from phospholipid precursors, that is arachidonic acid. Eucasinoids are inflammatory mediators, so they are important for inflammation.
Now, unlike histamine, which is made in the cell by the DNA and then stored in granules, eucasinoids are generated or made from phospholipid precursors during inflammation or for inflammation. The main phospholipid precursor that makes eucasinoids are known as arachidonic acids, which is a 20-carbon fatty acid consisting of 4 double bonds.
Okay, so arachidonic acid makes eucasinoids. Eucasinoids are inflammatory mediators. To make eucasinoids, arachidonic acid follows two main pathways by two main enzymes. It can either follow the cyclooxygenase enzyme or the lipoxygenase enzyme. If arachidonic acid follows the lipoxygenase enzyme, it will make leukotrienes, abbreviated LT. If arachidonic follows the cyclooxygenase pathway, it will make thromboxanes, abbreviated TX, or prostaglandins, abbreviated PG. So eucasinoids are the leukotrienes, the thromboxanes, and the prostaglandins. The thromboxanes and the prostaglandins are also known as prostanoids. In this video, we will look into the synthesis of these eucasinoids in a lot more detail, as well as their function. And also in this video, we'll look at some drugs that will inhibit the formation or the production and the function of these eucasinoids, and therefore suppress inflammation or halt inflammation. So here we have a cell. It can be a leukocyte, such as a mast cell or a macrophage. It can be an endothelial cell, such as the ones lining our blood vessels, or it can be cell fragments, such as platelets. Now, if we look at the membrane of these cells, the cell membranes are made up of a phospholipid bilayer. So here is a phospholipid. Arachidonic acid, what we're concentrating on, is made when a phospholipid is converted by an enzyme, phospholipase A2, PLA2, into arachidonic acid.
Phospholipase A2 can also convert the phospholipid to form another molecule called lysopaph, a precursor of platelet activating factor. So basically, the phospholipid can be converted to either arachidonic acid or lysopaph.
So we know that arachidonic acid will produce many inflammatory mediators such as prostaglandins and leukotrienes. So a way to prevent arachidonic formation is by preventing the enzyme phospholipase A2. During inflammation, the adrenal glands, which is above the kidneys, will secrete glucocorticoids or cortisol. So circulating glucocorticoid concentrations increase during inflammation as part of the body's homeostatic response, because glucocorticoids can convert to the active form, lipocortin. Lipocortin can inhibit the enzyme phospholipase 2, and thus the formation of arachidonic acid. We can also inject glucocorticoids, such as steroids for sports users, etc., etc. Now, glucocorticoids, as I mentioned, have powerful anti-inflammatory actions, but it also has powerful immune suppressive actions as well. So we should take this into consideration. Now, before following the arachidonic acid pathway and how it produces the eucasinoids, the prostaglandins, and the leukotrienes, we will firstly see what lysopaph does. Lysopaph will essentially convert to platelet activating factor. Platelet activating factor causes vasodilation, it increases vascular permeability, it also causes bronchoconstriction and works as a chemotaxis. So essentially, it promotes inflammation. Now, there are drugs out there that inhibit platelet activating factor activity, known as PAF inhibitors or platelet activating factor antagonists. So there are those drugs that can prevent this and thus prevent inflammation. Now let's follow arachidonic acid now. As I mentioned, arachidonic acid is important in making or producing inflammatory mediators. There are essentially two main pathways using two main enzymes.
The two main enzymes are lipoxygenase and cyclooxygenase. Lipoxygenase will essentially make the leukotrienes, while cyclooxygenase will essentially make the prostaglandins and also the thromboxane. Let's follow this cyclooxygenase pathway first. Arachidonic acid will be converted by cyclooxygenase to prostaglandin G2, which is very unstable and will quickly convert to prostaglandin H2 by the enzyme peroxygenase. So these forms, the prostaglandin G2 and H2, are unstable peroxides. So prostaglandin H2 will now quickly convert to a number, it can convert to a number of different molecules. It can, for example, convert to thromboxane A2 by the enzyme thromboxane A2 synthase. Thromboxane A2 causes vasoconstriction, platelet aggregation, and also bronchoconstriction. Before continuing on, we must know that the production of these eicosanoids varies between the cell types. So what I mean is that in our body, platelets will make mostly thromboxane A2. The vascular endothelial cells will produce prostaglandin I2, and the other leukocytes will produce a mixture of these. So leukotrienes, prostaglandins will produce a mixture of these. Now, the prostaglandin H2 can also convert to other prostaglandins, which share, some of them, similar structures. So for example, the prostaglandin H2 can convert to prostaglandin I2 by the enzyme prostaglandin I2 synthase. Prostaglandin I2, as I mentioned, is produced mainly by the vascular endothelial cells, and they cause vasodilation, they're hyperalgesic, and they also inhibit platelet aggregation. Hyperalgesic just means that there will be an increased sensitivity to pain, so we can see how it correlates with inflammation. Alternatively, the prostaglandin H2 can convert to prostaglandin E2 by the enzyme prostaglandin E2 synthase. This causes vasodilation and is also a hyperalgesic. Prostaglandin H2 can convert to prostaglandin D2 by the enzyme prostaglandin D2 synthase, and this causes vasodilation, and it also inhibits platelet aggregation. And finally, it can also convert to prostaglandin F2 alpha by the enzyme prostaglandin F2 alpha synthase. This causes bronchoconstriction and also causes myometrial contraction. So from these prostaglandins down the bottom, you can see how they have all similar function, and they all essentially promotes inflammation in a way. And so this was able to be produced. These prostaglandins and the thromboxanes, they were able to be produced by this enzyme mainly, cyclooxygenase. So what drugs or what pharmaceutical treatments is available that can prevent the function or the production of these? Well, we have prostaglandin antagonists, which work by inhibiting the function of these prostaglandins. They specifically inhibit prostaglandin F2 alpha, prostaglandin D2, and prostaglandin E2. Why does it specifically do these three? It's because these three share the most similar structures out of the four down the bottom here, because they only differ in the hydroxyl and ketone groups.